Hello, I'm Hyuk-jung Kwon. I'm at Bioinformatics Data Analysis Lab at Computer Engineering Department, and I'm also working at EDGC as a team leader of Bioinformatics team. Today, I'm going to talk about my research field and how this field is related to linear algebra. Since I'm still working at a company, my most of research field is related to our company work things. So today I'm going to tell about what's our research and what is our product in our company. So content looks like this. First part, GSA related service. And second, NGS related service, and third is AI related one. So this is first part. So yeah, this chip is the GSA, which stands for Global Screening Array. So this is manufactured by Illumina. Illumina is very well known NGS technology company. So you can see these many small sections. So one chip has 24 sections, and each section is for one samples. And each small section has 0.7 billion markers. These 0.7 million markers are very useful and clinically useful, or some healthcare, many things. We can analyze our disease, inheritance disease, or trait like Wellis strength, or some vitamin C, or blood pressure, or some things. So all these proven markers are designed to these markers. So in our company, we make lots of reports based on this chip. So, for example, the product name is Babygene. This is a newborn screening service. So we can find if some newborn baby has some inheritance potential disease or not. So you can see in this part of chromosome 22, the normal person, each normal person has two strands of chromosome, one from father and the other from mother. Yeah. So if someone has two strands as a normal, this plot looks like this. There is three bands. This band, and then in the center of this plot, we can see this band. But in this area, there's no band. That means there is only one strand. So we can find if there is some morbid critical genes here, then we can report this newborn baby has some potential inheritance disease. And also, we are checking the metabolic syndrome. The product name is Myzempren. So we can check our genotype data also, plus some health checkups. Every year we do the health checkup, and we can get some report from the hospital. So by mixing these two data, we can make this kind of report. This is the genotype risk, and this is the current metabolic score. And also we have another one, the risk care focus. So we found most critical genes, 25 genes, which are related to cancer, and this cancer should be some inheritance cancer. So if someone has a mutation in pathogenic markers, then those who have a pathogenic marker mutation, they will be likely to be an inheritance cancer in some age, not now. So they have some potential risk to have a cancer. So we can check and we can report this kind of things. Then if there is some critical mutation, they can consult with the clinicians, and they can go to see a doctor monthly or every 3 months, and then should check if there is any grown cancers or not. And second part is NGS. NGS means new generation sequencing. So by this new generation sequencing technology, we can easily get our whole genome data. So in our company, we are doing major two kind of things using NGS technology. One of them is NICE. NICE means our company product name, and this is the NIPT, non-invasive prenatal screening. Okay, that means when a woman is pregnant, if they have more than 10 gestational weeks, in mom's blood vessel, there are more than 10% of a very small fragment of fetus, not all from moms. So we can check this very small amount of fetus blood. We can check this fetus has Down syndrome or some other Patau syndrome or some things. So this is one of our major product and major research field. And the other one is liquid biopsy. So liquid biopsy is the same as NIPT. In NIPT, we check the fetus's cell-free DNA. In liquid biopsy, we check the ctDNA. ctDNA means cancer cell-free tumor DNA. So if cancer cell is growing, there are three types of phenomena. It happens. One is apoptosis and necrosis, and the last one is secretion. So by this mechanism, there are some cell-free DNAs from the cancer cell. So if we can check this cell-free DNA which are popped out from the cancer cell, so we can predict if there is a cancer is growing or not, and also we can check what type of cancer is growing before we realize it. So if this liquid biopsy service is prevalent in near future, then so I think we can say we already overcome the cancer, because we can find what cancer types of my body before it is detected by some other device like a sonography or something, X-ray or something in clinicals. Yeah. So if someone should get a check from a hospital, they should have some kind of symptoms of cancers. So once a doctor decide to biopsy and check if tumor is really cancer or not, but they should have to they should do the biopsy. But with this technology, we can just gather a blood, and then we can check if there is a cancer is growing or not. It's very useful, and at the same time it is very, very difficult to make a service. So currently our company's research team is strongly focus on this liquid biopsy technology. Okay, so we can see another things. I already told you that there is 2 copies in every chromosome in every person's. But if there is a cancer, the cancer cell is growing out of control. So some part or some chromosomes have more than 2 or 3 copies. Sometimes we could see more than 10 copies of one chromosome. So in this sample, this is a lung cancer samples. The red means there are high number of copies, and this light color it looks like a normal. But in this chromosome 3, the upper part, front part has missing. So this is just 1 copy, and this part has more than 3 copies. So you can see this is a normal samples, and this is a lung cancer samples. So we can easily see this lung cancer sample has so many CMVs. So by checking the ctDNA and cfDNA, you can see these kind of things. Okay, the last part is AI-based technology. Okay, so sometimes we are doing many research based on our GSA chip. GSA chip has more than 0.7 million markers. With these markers, we can make lots of things. We can predict migration of some ethnics. We can predict some height, like if someone has some genotypes of certain genes relate to height, then we can predict how high will he or she will be. And we are sometimes when we make some clustering, mostly we are using k nearest neighbor. It is called KNN. So I think this part is my research field is related to linear algebra. So actually we don't calculate very deep formula in linear algebra. Most part of we are making regression or logistic regression or linear regression inside of function. The main calculation is all based on linear algebra. So, and then we are also researching on the deep learning, and actually deep learning is based on the tensors. So if there is a tensor 2, we call it matrix. So most of part of each deep learning layer, so each node has a very simple calculation, like y equal to wx plus b. W is weight and b is bias. So this wx, this calculation is occurred in many nodes. If there is more than 10,000 pictures, we will have 10,000 pictures of one layer. So to train properly, sometimes we use GPU chip, and sometimes we only use a CPU. Whatever we are using, we need a very fast calculation of wx plus b. So this is based on the linear algebra matrix multiplication. So I think this AI, deep learning or machine learning field, is tightly related to linear algebra. So if someone knows better in deep part of linear algebra, so we can easily understand how the deep learning and how the picture selection is working. So I think this is a very important things to study linear algebra. So I think this semester I choose linear algebra, and although it was kind of difficult to understand all of things, but it could be a good motivation to study linear algebra more deeper than before. So with these AI techniques, we can prediction of drug adverse effect. Also, so we are also doing this one. This is the last part of my presentation. And this is the phenotype prediction with the genotype. Actually, this phenotype is the skin phenotype. So we have many phenotype data measured by 10 different devices. So using this phenotype data, and we are going to calculate or match the how much correlated with the genotype. So to assess this model, we first we selected markers by some method, and then we did the machine learning. So most of this is the linear continuous number of data. So we use the regression. So this part we use the linear and it's lasso elastic, and this kind of decision tree. So we did the K-fold cross validation by taking the 80% or 20% as a test. So we found one of the phenotype, this is alpha. We could find more than 63% of accuracy prediction with the ridge regression. Because this is the ridge regression, this is the phenotype, and this is the prediction. So we could see with the genotype, we could predict quite accurately. So, okay, this is what I prepared for this presentation. So, like I told, all our research should apply machine learning or deep learning, as we get many samples, but sometimes we need much more knowledge about the mathematics behind all the modeling tools. So without this one, anyone can manage to do that. But if someone knows better about the deep inside of linear algebra, so we can see find the result much better and much easily. So I think this semester's this subject was very useful, and it has a good motivation to me. Okay, thank you for listening.